so I guess I'll get my formal introduction tomorrow, but my name is Allison Hsiang. I'm a researcher at Stockholm University. I'm a paleontologist. I call myself a computational paleobiologist, whatever that means. But I work in deep time, basically. And so today I'm going to talk about sort of my experience building image datasets and just some tips and tricks that I've learned that hopefully will help you avoid some problems when you build your own datasets. So this is mostly coming from my experience building this dataset, Endless Forams Most Beautiful. So this is a dataset of modern planktonic foraminifera. I'll talk more tomorrow in my keynote about what planktonic foraminifera are, what they're used for, why they're important, and all that stuff, and what we learned from this dataset. But all you need to know for now is that this is a dataset of about 36,000 modern planktonic foraminifera individuals. And so it's split between this Yale Peabody Museum dataset and this dataset from the Natural History Museum in London. And we use this dataset, which contains a total of 34 species. So for reference, there are about 50 modern planktonic foram species. So 34 species represented, and we use this to train a deep learning model, VGG16, to get, I think it was 87% accuracy on identifying these species. So this was our pipeline for generating our data set. So basically we have our microfossil sample, we image it using a high throughput light microscope system. We get these Z-stacked images, then we segment these Z-stacked images to get our individual objects. So then we can use focus stacking to get our nice focused image of each individual, and then we attach a species label. So we can break this up into sort of the image acquisition part, which is most of this pipeline, and then the label acquisition part. But actually, I'm going to talk mostly today about the label acquisition part and a little bit about the image processing stuff. But the imaging stuff is much more, specific to the type of organism you're looking at, the type of problem you're trying to answer, so I won't talk about that so much. So yeah, the image acquisition, label acquisition comes together to produce your training set. So we've already seen figures like this several times before during this forum, so I won't go into specific detail, but I just want to make the distinction here that I'm going to talk about classification problems. So in classification problems, you're just saying, is this class that I've defined in this image? So generally with this taxonomy stuff, we want an image that has a single specimen in each of these images. Versus instance segmentation, where you're doing sort of the localization in the image along with the classification. So you don't need to break them up into individual images. You can have one image with multiple specimens in it. We're going to talk mostly about this classification problem. So the way we built this Endless Forams dataset is we wrote a software package called Automorph. So this is a open source software package. It's written in Python. Which you can, so there's a published version, there's a paper associated with this. You can find the published version of this software at, so I was in Pincelli Hull's lab when I worked on this. So the whole lab GitHub page. Since then, I've also done a lot more development to Automorph, so you can find the latest version in the development fork on my GitHub page. The latest version there's like a Docker version, there's like a easier way to install it using conda packages and so on. So if you're interested, you can take a look there. But Automorph is basically built for doing this problem where you have an image that has multiple specimens on it, and it will basically go in there, find where all the individual objects are, crop them out, save them individually, and what it also does is actually do sort of simple morphometric measurements of all of these objects at the same time, so things like major, minor, axis length, perimeter, area, and so on. So you can get all that in a single easy step. So it does this not using deep learning or anything fancy, just using sort of traditional image processing algorithms. So you're basically just looking at the values of pixels in the images and then running, applying a series of filters to find where the objects are and get where the outline is. And then also if you have Z-stacked images, then you can get sort of 3D information by getting the heights of each pixel, each focus pixel on each of your Z-stacks. So of course there are other ways that you can do this. ImageJ and Fiji is a very popular software package for doing this kind of stuff. The difference here is really, so we built Automorph because we wanted to really deploy this at scale. So to be able to sort of have as little manual interaction as possible, be able to run it on a high-performance cluster, be able to run thousands and thousands of objects and extract this information without having to mess around with it so much. But of course, the problem is that if you're not so familiar with the command line, for instance, there might be sort of a learning curve before you can really use automorph so well. So then VG and ImageJ has this nice GUI and whatnot. So it really depends on what you need to sort of decide what is worth it for you time-wise. If you're not familiar with the command line and maybe you only want to annotate a few thousand objects, a few hundred objects, then maybe it's not worth it for you to use something like automorph. You can use something like ImageJ instead. But if you want to do hundreds of thousands of objects, then you might want to pick something more like automorph that can be deployed at scale. So the first thing, so that was the image processing stuff. So now we'll move on to the labeling stuff. So this is an example slide from the Endless 4Ms dataset, and what you will see is that there are many different types of objects on here. So we basically took our samples, we cleaned the samples. These are core top samples. You drill into the ocean sediments, you pull up all the dirt, and then you clean it and you look at the objects that are inside. And so what you'll see is that there are 4Ms here, but there are also things that are not 4Ms. So these like really big shiny things here, these are other things. There are things like rocks, there are pieces, there are fish teeth, there are spines from sea sponges and so on. And so our first task was this really sort of coarse classification task, where we just wanted to separate out, the stuff that is 4Ms and the stuff that isn't 4Ms. So for this we wrote, sorry that the slides are all offset, but for this we wrote like a very simple Python program called classify specify. So this has a GUI. This is just like the Python tkinter, very simple, lightweight GUI development. And so the idea here is basically you can load in a folder that has however many images, and what this program will do will basically show you each image. You can click on what category it belongs to, the confidence you have in that, and then it'll automatically bring up the next image, and then you get spit out this CSV file that has all of your classifications on there. And then if this is, if you have a lot of different classes, you can also say load in a list of species. Maybe you have 50, 100 species, and you don't want buttons for all of it. So then you can use this text box instead. It has autofill or whatever to allow you to sort of collect these species labels quickly. But the coarse classification is a much simpler problem. So this, is the type of thing that you can teach other people to do relatively quickly. So we had undergrads work on that. But for the for the actual species identifications of the planktonic foraminifera, this is a much harder task. So this is a task that's kind of unavoidably, you need to have taxonomic experts participating in this, because it would just take way too long to teach people how to differentiate between these species. I'm sure some of you before this talk have never even heard of planktonic foraminifera. I hadn't heard of them before I started working on them. So, having to teach people to make these species distinction is just something that takes too long. So we wanted something that could allow us to bring together a large team of taxonomic experts to collect species labels for this project. So we ended up using
I'll fix it.
So we ended up using Zooniverse, which we've heard a little bit about from other people. So this is a citizen science platform, and
This is good.
Great.
Sorry about that.
No worries. So yeah, this is a citizen science platform, but you're able to make private projects on there. So that's what we did, because we didn't want random people to come and start labeling 4Ms, which would probably be incorrect. So we uploaded 40,000 images of planktonic 4Ms onto this private project, and we collected a total of more than 140,000 identifications. Now you might wonder, there's only 40,000 images, why did we have 140,000 identifications? And the reason for this is because we really wanted to make sure that the quality of this label data set was really good. As has been brought up earlier about how data quality is really important, what we really wanted to make sure was that everything was as correct as it could be. So what we did was have every image be identified independently 4 times. So this is quadruplicate classification. And the reason you want to do this is because it gives you a way of telling which labels are actually good and which are bad. So in this example here, we have this one image. So we have 4 people who've looked at it. 3 of them say it's Globirotalia monardii. 1 person says it's Globirotalia ungulata. Now imagine if you only had 1 person label each image. If this person had gotten to this image first, you're going to have an incorrect label associated with this image. And you're not going to have any way of knowing that unless you go back and look at everything, which you don't want to do. So in this process, at the very least, you can, we took the threshold of saying we only will accept something as a high quality label if at least 3 out of 4 people agree on what it is, and everything else we just throw out of our data set. But then what you can also do is go back and look at the ones where only 3 out of 4 people agreed and look at what that 4th classification is and also draw some conclusions based on that. So these were the accuracy rates for our 20 or so taxonomic experts. And what you'll see is that the average human accuracy of our experts, here we're assuming that the label where there's 75% agreement at least is in fact correct. But based on that, we have an average human accuracy of 71% and a range from about 64 to 85%. And this sort of levels of human accuracy are actually pretty standard. If you look at other studies that have looked at this specifically, that is how good are humans actually at this sort of repeated taxonomic task, you'll find that 60 to 80% is about what you see. So this is another study looking at planktonic foraminifera. You can see that the accuracy overall of all participants, so they split it between experienced labelers and students. So experienced obviously are better, 78.5%, the students 57%, overall 59%. So this is kind of in line with what we were saying. This is another study looking at pictures of bumblebees, and this was actually a contrastive test, so they were looking at, look at these 2 pictures of bumblebees, are they the same species or not? And what you can see here, so here they're looking at people who are experts in bumblebees, people who are not experts in bumblebees but who do work in conservation, and then people who are not experts and who do not work in conservation. So the conservation is the middle gray and the non-conservationists is the darker gray. And what you see is that that actually they don't get that many correct, right? So they're getting somewhere between 50 and 60%. This one down here is consistency. This is basically looking at if you show people, the same person, the same images and ask them to do it multiple times, do they actually give you the same answer? And the answer is not always. So this is really important to see because it basically tells you that even with experts, you're going to have 10 to 20% of your labels are just going to be wrong for whatever reason.
Am I reading that correctly? The people who think they're accurate, so the middle gray and the bottom, they consistently think they're less accurate, or less than non-experts?
So this one...
That's an amazing result. Is everyone seeing that?
So yeah, so this is consistency, just like you said the same thing.
Yeah, so the people who think they know what they're talking about, but they are not experts, are consistent, but they're actually less accurate. That's incredible.
Yeah, I mean, there's yeah.
Oh no, that is incredible. I was gonna ask if this is aggregated across species. Like, so one of the things that we've found is that there's a great ecological paper about this recently from Lund that basically says these are the sets of small mammals that no one should even try to ID from images because experts cannot agree. It's basically just stop telling me you're identifying these from images. As a human, as a machine learning model, I don't care, no one can agree.
Yeah.
And so I'm curious if it's do you have some species that are actually identifiable and some that are just not identifiable, or is it kind of uniform across the board?
I don't know about this study particularly. In planktonic foraminifera, it's kind of an interesting thing because there are multiple taxonomic schools about how you actually... so, I mean, yeah, so there are lumpers, there are splitters, there there's a lot of gray area between where these species boundaries actually are. So yeah, that is another problem. Yep.
Hey, yeah, thanks. Really interesting. Maybe you're going to talk about that, but the fact that you have, assessment of intersubjectivity, it sort of denotes that maybe certain images are more ambiguous. Do you or can you explicitly model the ambiguity of each image rather than, I don't actually care what it is, I want to know to score the ambiguity now. And you have ground truth data because you have multiple.
Yeah, so that's something we're thinking about. We haven't done that yet, but it is something, because we have this data where we can sort of quantify that ambiguity.
Grant Van Horn does some work on like aggregating labels across people and computer vision models that explicitly models how challenging images are as like part of the system, and like basically, it does seem to help getting sort of more accurate like agreement, like combined annotations out at the end.
And we also, for the course classification stuff, we did have people sort of like self-evaluate how confident they are in their labeling, but of course that's very dependent on the personality of the person, like the mood they're in, that sort of thing. Like you might have someone who's, we'll get to that, I guess. So yeah, it's important to realize that human taxonomists aren't perfect and they make a lot of mistakes, and that's why you need to do stuff like quadruplic classification so that you can actually figure out where the problems are and deal with them. So another thing that I like to point out is that when you collect labels, you should not give your classifiers an option to say, I don't know. And this seems, a lot of people, you tell them this and it seems kind of counterintuitive, and the reason you do this is because by allowing people to say, I don't know, you're effectively throwing away a lot of information. So in this example here for this image, so person A has said this is Globirotalia monardii, person B says Globirotalia timida, C says Globirotalia ungulata, and D says Globirotalia monardii. So there's not agreement here. We wouldn't consider this a high quality label, but what we can get from this information is that very likely this thing belongs to the genus Globirotalia, and that is information that you can potentially use later. Whereas if you allow people to say, I don't know, then you might get one person who says, well, I know it's monardii. Everyone else says, I don't know, and you've lost all that information, because now, again, you only have a single person who said anything, and you can't check whether that's correct or not. So that's, and different people have different thresholds for saying, I don't know. Someone may actually, in fact, be a very talented taxonomist and be very good at species identification, but not have a lot of self-confidence, and they might just say, I don't know, at a lower threshold than someone who is not very good at it, but is very confident. Yes?
Yeah. Have you tried to get like confidence ratings from participants so that then you can kind of weight their
Yeah, so we didn't get it for the Endless Forams database. We did get it for that course classification task that I mentioned before, but we haven't gone in and looked at. We haven't evaluated things yet. Yeah, unfortunately. But yeah, so this figure up here is from that bumblebee study again, and you'll see that there's some 10 to 20%, around 10% of people will say I don't know. They had that option. And what you actually see is that the experts actually say I don't know much more often than the non-experts do, because the experts know more when they don't know something. And then this here is an example from, so this is from Snapshot Serengeti, which some of you have probably heard about, where they had these camera traps in Serengeti. And they were trying to get, this was a citizen science project where they got people to sort of look at these images and identify what was in them. And this is actually from their blog post where they talk about why they don't allow you to say I don't know. And what they say is that for this image, clearly it's very hard to see what that is. And they collected, all these sorts of different labels, but what they could conclude is, well, this is probably a small mammal of some kind, right? No one's saying it's a rhino or a lion. So that is some information. Everyone said it's moving, so this is the type of thing that you can get by not allowing people to just sort of throw their choice away. Yep.
This is a naive question from someone who doesn't do any of this, but I wonder why, was it worth the investment to identify some subset of these things and then do like, I don't know, SEM or something, or in this case like trap a creature and then let it run in front of the camera a bunch of times. I mean, just like some like very high quality, incredibly expensive ground truth to kind of take these sorts of, I don't know, and like map them somewhere into some knowledge space that you have some information about. Because I think there's one question which is like, it's literally unknowable what this thing is, or it's like it's a blob that's like a mutant, and it's like no one can know what that thing is. And the other, and you want to distinguish that from, actually, it's knowable with the right training and with the right expertise, and it seems like unless you have that high res, or whatever, insanely high quality, you'd never know. Is that ever worth it?
I think it can be. So I will actually talk a bit in my talk tomorrow about
Then you can save something if you want.
about SEM and how that kind of plays into this kind of stuff. Yeah, so sometimes, maybe, is the answer. Yeah, so another thing that kind of ties into what Ben was saying earlier about knowing your data set and really getting in there is standardization, which is you need to sort of be very clear about what the boundaries of the different classes that you have defined are. So like I said, with planktonic foraminifera, this is a problem with the different species boundaries that people draw. So for that project, we actually specified a specific taxonomy and told people to follow that. But there are also things like, for instance, we, in our course classification, we were separating between things that were complete planktonic foraminifera, things that were broken, fragmented. This is important because fragmentation rate of planktonic foraminifera is actually a really important thing that people measure through the fossil record to figure out things about what was the environment that these things were living in like, and what was the preservation regime like, and so on. So then, if you have the category damaged, complete, where do these types of things fall in? So this first image up here is a complete foram that's nestled in the broken shell of another foram. So, you need to make sure that your classifiers know what to do with that. The second one, it's mostly complete, but it's got this little tiny hole in it. So it's technically damaged, but it offers pretty much all the information you would need from a complete specimen. And then this last one is, is mostly complete, but there's a tiny bit on the bottom right hand there. There's a tiny bit of actually a chamber that has broken off that you can see. So these are just things these are things you can only know if you actually understand your data set, understand what's in your data set, and know what these edge cases are so that you can define which categories they actually fall into, so you can be consistent about how you actually label them. And so this is sort of specific to, of course there's intraspecific variation in pretty much everything, but in planktonic foraminifera it's particularly acute. So you can see here, this is one species, Globigerinoides ruber. This is just an example of the type of variation that you'll see within this one species. And so you need to do things like provide field guides, provide taxonomies for your classifiers to follow. And this is standard. This part is just talking about standardization in terms of also organizing your metadata well. So we had a lot of metadata associated with where these guys were found, where in the sediment, stuff like that. So just make sure that you're very clear about all that metadata and organizing it in an understandable way. So when you're building these large data sets, you need to get people to spend a lot of time doing this annotation. And especially if you need taxonomic experts, these are people who are, have been working a long time. They probably have better things to do with their time, but you want them to, contribute as much time as possible to your project. And so one thing we realized is that we would send out a weekly leaderboard of how many identifications every person had done in the project. And we found that this was actually very, very useful. I had multiple people contact me and say, I just really want to beat this one person. so they would use this, to foster a bit of friendly competition to get people to. Yep.
You also point fingers at people who are widely different from others.
Yeah, so we anonymized it. We sent it out, but some people claimed they could tell who was who. Yeah, so scientists tend can sometimes be competitive, so you should use that to your advantage. There have also been other studies that have looked at various ways to sort of encourage people. This one here, so this upper figure is actually looking at the difference between people who are volunteers and people who are paid. So they hired people via Mechanical Turk, and they compared that to volunteers and looked at how many images did each group annotate. They also tried out this feature where they tell you how many images you've annotated out of the total number of images. And they actually found in this study that the people who are hired had consistently lower accuracies than people who are volunteers, which makes sense because they care less about perhaps the success. And this down here is just looking at another study looking at, what's the best way to sort of advertise your project if you're trying to get, random public to join in. And they found that overall email lists were the best way to do this. Much, much better than any social media advertising or so on. Yes?
Yes, I was wondering about this labeling, labor-intensive labeling issue. What are your thoughts about unsupervised learning, also as a way to maybe limit the number of labels that you need, and maybe even, I don't know, possibly increase the generalizability of your algorithm, so you can use it maybe for one species, but maybe actually can generalize to another one or something?
Unsupervised unsupervised algorithms are something that we haven't looked so much into yet. It's of course on the wish list of things to try, but so far everything we've done has been in a supervised paradigm.
It's complementary, it's not a replacement. Like, you still need real labels for everything, but you might be able to get a better starting point using self-supervision.
Okay, so so that was all stuff dealing with sort of this classification task, and for the last bit here, I'll just talk a little bit about instance segmentation, which is a bit more of what I'm working on now. So there are, of course, lots and lots and lots of different tools that you can use to do this sort of annotation. There are lots of proprietary tools. There are also lots of open source tools. I use segments.ai for my work right now. It's proprietary, but actually there's a free educational license where you have unlimited image uploads and so on. But just some things to think about when you're trying to choose what program, just because there's so much choice. So there are questions, how do you choose which program to use? So first is, of course, what is the cost? So how much grant money do you have? Do you have any grant money? This can depend on status. So like I said, there are educational discounts and so on. There are some programs that will, that are proprietary, but they say you can, upload 1,000 images or something like that. So that might be sufficient for what you're looking at. And also some limit it by, these features are free and these features are paid. And then another thing to consider is what type of data do you need to annotate? Of course, are you looking at 2D images? Are you looking at 3D models? Are you looking at videos? Does it have any additional features that make it particularly easy to annotate the type of data that you're looking at? So some of these programs have, for instance, shape interpolation between keyframes and videos that can make it easier for you to annotate an entire video, all the frames. What different ways are there to create annotations? So this one I put in because I really hate drawing polygons around things. It's like my least favorite thing to do. So this was really important for me. So there are you can draw boxes, there are polylines, points, freehand. There are lots of programs that have sort of automated ways of sort of predicting where the objects are and so on. And how does that square with the hardware I have available? So by that I just mean do you have access to a tablet, right? Because that can make annotation much, much easier in certain ways than in others, than if you only have a keyboard and mouse, for instance. Does the program have a sane interface that I work well with? Often people have recommended I use things and I use them and I hate it, so you have to sit there for hours doing this, so make sure it's something that you actually like using. What data formats does the platform support? So, you know, if you're using certain models, you know, they have a certain input type of data format. So you want to, you know, maybe if you don't want to mess around with converting things, you might want to think about that. And then what features are there for quality control? So for instance, how easy does this program make it for you to have multiple people go in and look at the same annotations, and so that you can actually do this multiple classification of the same object thing to make sure that your labels are actually high quality. Yeah, and then at the very end here, these are just kind of random things that I thought of while I was making this talk. I don't know, you know, what everyone's experience is, but this is just that, you know, I work with paleontologists, and the level of computational understanding is perhaps not as high as in other fields. So there are just things that you don't think about until you start doing it, and then you realize, oh crap, like now I have to go back and fix a lot of really annoying stuff. So like things like text encoding, like if you're doing a lot of string processing, and then you figure out that the way you're processing it doesn't play nice with, you know, your Windows formatted text file that has a different way of encoding like an enter key. Things like putting some thought into designing your file name, so use these separators deliberately. Make sure you don't mix them up, because when you are processing hundreds of thousands, potentially millions of files, you want to make sure that you don't, you know, your code doesn't break in a loop or something because you accidentally switched a hyphen and an underscore or something like that. And also make sure that, because sometimes you need to separate things based on, you know, this block of information that you've encoded into the file name. And so that's something that you need to think about and design before you start saving images. Learn how to use shell scripting and tools like regular expressions. So this is again having to do with processing tons and tons of strings, tons of text. Regular expressions just make everything easier. And plan your pipelines to minimize manual labor as much as possible. So this isn't just to avoid having to spend time to do manual labor. It's also because when errors, so most errors occur during manual data editing. And so if an error occurs, you have no way of sort of retracing your steps. You just have to kind of, you know, you happen to see that you made a mistake somewhere. If you copy and paste the wrong thing into the wrong spot, you have no way of finding that. So having these pipelines, you know, minimizing the labor as much as possible, having explicit processing pipeline with all your commands, including all the commands you use to process that you throw into the command line, is really important for catching errors, and also for this reproducibility aspect, that someone else can go back there and just run your entire pipeline from beginning to end and get the same thing out. Yeah, so these are the same things, like plan to optimize your data structures, and use version control, I've said before, GitHub, and practice effective code commenting. So going back again to the thing about how your worst enemy is yourself, basically. I've written code so many times that I go back to a few months later and I'm like, what was I doing here? I have no idea. So this is really important, yeah. And to say nothing about giving that code to someone else. Like, they're not going to be able to do that if you can't figure out what you're doing. Yeah, so that's all I have.